Our next presenter, Robert Wimbish, is a fourth-year PhD student in biochemistry and molecular biology. He shared a lot of information with us about who he is and where he's going, but I found it interesting that he started college as a rugby-playing sports management major with aspirations of managing a major sports team. But he took a science class and realized he was passionate about the natural world and transitioned into training as a chemist. And then he learned more about biology and decided he wanted to study more complex living things. So he transitioned away from organic chemistry, good move, and into cell biology. So I'm fascinated. His title today is Attachment Issues, Uncovering the Molecular Basis for Chromosome Movement in Cells.
Each of you started as a single fertilized egg, and through the process of cell division, you're now made of over 37 trillion cells, each of which contains a copy of your unique DNA, the genetic instructions manual, which makes you who you are. Dividing cells package their DNA into structures called chromosomes, and they move their chromosomes using a football-shaped machine called the spindle. These are images taken on my lab's microscope of real cells dividing. In these images, you're only looking at the chromosomes in red and the spindle in green. What you'll notice is that the spindle has to form direct attachments to the chromosomes. If it forms the correct attachments, it can then generate force on the chromosomes to pull them apart towards opposite ends of the dividing cell. If it does this correctly, then the two new daughter cells that form will each have a full complement of identical genetic material. So if you imagine me as a chromosome, and each half of me is an identical copy of DNA, the spindle has to physically link up with each half of me and pull me such that my right half will go into the cell on your left, and my left half will go into the cell on your right. This is the ideal scenario, but we know that errors in this process can occur. For example, both halves of me could hook up to the same side of the spindle. If this were to progress, my entire body would be pulled into this cell, leaving this cell without a copy of that chromosome. This can be catastrophic for a cell, this process called chromosome missegregation. It's akin to you trying to build an IKEA furniture set with an instruction manual that's missing half the pages. We know that tumors, 90% of them show abnormal chromosome copy numbers. You can see examples of this with a healthy cell that has 2 copies of each of its chromosomes compared to a cancer cell that has multiple of certain copies and is missing entire ones altogether. My project has been focused on understanding how chromosome missegregation events occur. Specifically, I've been using high-powered microscopes to look at the attachment points between chromosomes and the spindle. If we can better understand how these attachments occur, we can better understand how they go wrong in cancer cells. I've been using a combination of genetic approaches and chemotherapeutic drugs to tweak the attachment strength in human cells. Through this, I've uncovered new molecular details about the timing in which these attachments form, their composition, and the precise mechanism that a cell uses to regulate their strength. The strength of these attachments is particularly important. If they're not strong enough, the chromosome won't be able to hang on to the spindle and won't be able to move with it. And if they're too strong, it won't be able to reverse incorrect attachments like the one that I talked to you about earlier. It's my hope that these experiments will provide insight into how incorrect attachments are formed and can lead to devastating diseases. But overall, I believe my project will provide a more broad understanding of how everyone in this room made the journey from 1 to 37 trillion. Thank you.